Good morning, everybody. Thank you for coming back for our second day of Lollapalooza. It's going to be just as much fun as day one, if not more so. Quick reminder, the beverages and Tootsie Pops are over there. Restrooms on the northeast and southwest corners of the building, also water bottle fillers. 2 morning sessions, followed by lunch. Lunch is scheduled from 1 to 2:15, and it's not served here. So attach yourselves to some people who know their way around, or go foraging on your own. And then we'll come back. We've got 2 afternoon talks. And then at 5 o'clock, we're going to have a reception and make some toasts and have some recollections and stories in Bob's honor. Honor. Yeah. I need to say thank you, of course, to the sponsors for the event, which is the Department of Philosophy, the Office of Research, the Taft Center, the College of Arts and Sciences, and thank you to our colleagues and Bob for making this possible. Jackie mentioned yesterday that some of the people here, including Bob, were participants over a series of years in what she called, as I recall, the multiple realization circus. Traveling circus. That's sort of accurate. So it was started by Larry Shapiro, who sends his regrets because he couldn't attend, having recently had meniscus surgery. So if he's watching the stream, sorry, Larry, we're having fun without that.
I heard some really good drugs.
Yeah, good drugs? Okay. So after a while, it seemed like I should invite people here for the traveling circus. And so I went to the dean, who at the time was Valerie Hardcastle. And this is the bad part about having a philosopher as your dean. I went and I said, can we have some money to have this workshop? And she said, I'm not going to give you money for you and your friends to sit around and talk about philosophy and drink beer. And so, okay. So some time passed, and she came back and she said, actually, we have a donor who gave money for faculty development. Would you like money for you and your friends to sit around and talk about philosophy and drink beer? I said, sure. So time went on, and I was department head for a while, as some of you all know. And as I was looking towards the end of that, I thought, it would be nice to have another event. And I was talking to Bob, and I said, how are we going to get the money for this? And he said, well, I could just retire. No, you don't have to do that. But he insisted. So here we are. So thank you, Bob. And again, thank all of you for being here. And now Steve is going to introduce our speaker for the morning, first speaker for the morning. So you bet.
Hi, everybody. Hi, Bob. Sorry I'm late. So I'm Steve Downs from the University of Utah. And before I introduce Bill, I'm going to say a couple of things about Bob. So Bob gave me my first job in philosophy. Cheers, mate. He's probably regretting it now. And I have one lovely story. So he was a department chair when I was hired here. I was hired on 1-year visiting positions. And this is before you could zoom into your classes, before we could accommodate. And I phoned Bob up from West Virginia. The car's broken down. I'm in a panic, beginning assistant professor. Can't teach classes. I can't drive from West Virginia to Cincinnati because the car's broken. There's no public transport because we live in the USA. So I'm terrified. And I phoned him up and I said, I can't teach class on Monday. He's, no big deal, we got it covered. So, thank you. Thanks very much. And it's the 30th year anniversary of discovering complexity. Bill Bechtel. So I think it's another nice reason for this occasion to be happening now. And I'm just going to turn it over to Bill, who's got the stage.
So a word of warning as I start this talk, I do not respect trademarks or copyrights on the term mechanism. It's in the public domain to be used as works for various purposes. Going back not just 30 years, but 40 plus years, when I was working across the river at Northern Kentucky and Bob and I were beginning collaboration, we set out for a project. But it was not a project on mechanism, and we did not set out to write Discovering Complexity as a book about mechanism. It was rather a project on trying to understand the philosophical conception of reduction and whether it applied at all to the sciences that we were dealing with, biochemical genetics, biochemistry, and so on. And we encountered a number of problems, but one problem that really stood out is that we didn't find theories presented as sets of propositions, which we could step into a deductive relationship and show how one was derived from the other. Nonetheless, the research we were looking at sure seemed reductionistic in the scientist sense of the term. Now, instead of theories, we discovered that the scientists referred to mechanisms, and they offered mechanisms as explanations for phenomena. So that's where we turned our attention. How do scientists develop mechanistic explanations, mechanistic accounts? And as you're all aware, one of the ideas we developed was, well, you make certain assumptions that you can decompose the system as a heuristic. You can try to identify component activities within the system. So so that's the structure that came to be Discovering Complexity. We took the word reduction out of the book, except for two places, one of which was to tell Marjorie Green where the other occurrence was. But it sure is a book about reductionistic research. We, I think, fulfilled our mission to the National Endowment to the Humanities, who funded the project. But flash forward, well, since its publication, almost 20 years, that 2008, 2009, we were, Bob had arranged for to have it reprinted by MIT Press. We were preparing a introduction to the new edition, and the landscape had changed dramatically. You didn't find much talk about deductive relations between theories, but thanks to Carl and his collaborators, Lindley and Peter, to Stuart and others, the notion of mechanism was certainly in play, and people were talking about mechanistic research and the strategies by which mechanisms are discovered. I think all of us grounded our approach to mechanism in what we found in the sciences. This was not an a priori concept, but rather a concept that was being used by the scientists. And so we tried to understand what they meant by mechanism by looking to what they did with the concept, how they used it in their scientific work. Now, each of us have published various versions and accounts of what we take mechanism to be in those contexts. But I have to say, surely, the cases we looked at turn out to be selective, and they are not, there's no reason to think they are representative or that there is even a body to be representative of. And it seemed highly likely to us, and we emphasized this in the introduction to the second edition, that if one went out and looked elsewhere, one would find different ways the notion of mechanism is being used, different ways people are investigating them, and we'd learn a lot more about what scientists mean by mechanism. So what I'm going to do today is briefly describe three such journeys I've been on over the last 15 years or so, and different ideas about mechanism that have come to me from these journeys. And let me caution, I have no sense that we're even beginning to approach comprehensiveness at this point. But these are three ways that I've come to think about mechanism differently from looking at different sciences. So I spent a long time in the circadian world And one of the themes I'll draw from that is the incredible importance in many cases of computational modeling in order to understand how mechanisms behave. I will then turn to some more recent work I've been doing on molecular motors, and a stunning realization one day that I've been talking about mechanisms doing things for years and never asked, well, where's the energy that figures in the account? So then the role of understanding the constraint of free energy is what mechanisms are doing. And third, I'll turn to some other recent work, getting beyond the cortex and looking to other parts of the brain and asking how are they organized. And I would introduce the idea that it's certainly not hierarchical in that context. It's what I refer to as heterarchical. So let me start with the circadian world. Here the story starts very much like the story Bob and I told in Discovering Complexity, pinning down a phenomenon. By 1960, there was a fair amount of agreement amongst people working on biological rhythms that organisms had an endogenous approximately 24-hour rhythm that was temperature compensated and entrainable, but there was no clue as to what was responsible for that rhythm. So the challenge was to identify the mechanism. In rather heroic work, Ron Konopka did some genetic screening in Drosophila, found a gene, called it period or PER, that when mutated produced altered rhythms, either faster or slower rhythms, or sometimes arrhythmic fruit flies. And we had a first possible component of a mechanism. 15 years later, when cloning was a possibility, Paul Hardin, together with his future to be Nobel laureates for this work, Paul didn't get it, came up with one they could show the timing of the expression of the mRNA and the protein and showed that they were in a phase relation, but moreover, proposed a mechanism. That is a way of putting things together to create 24-hour rhythms. Now, something to note about that diagram. For the few icons on it, 5 of them are question marks. Basically, everything's a question mark. The idea is that is there. Somehow, a feedback loop is going to generate oscillations. What's fed back and where it's fed back to, don't know at the time. Now, good reason for thinking it could generate oscillations. Engineers had known for a long time that negative feedback produces oscillations. Question, though, is we're trying to understand something that's rhythmic intrinsically, is are these oscillations going to flatline eventually, or are they going to be sustained? Have we got a potentially sustainable oscillator? And the problem is just staring at what you think you've got, a particular protein that you think in some way is inhibiting its own expression, is not going to tell you whether you've got something that could sustain oscillations. So Al Goldbeater picked up the baton at this point and said, well, let me write some equations to characterize what you guys at Brandeis had come up with, a feedback loop. So first equation, that's just the first equation in it, tells you how tells you about the buildup of the concentration of the per mRNA and then its degradation. Wrote other equations and to characterize, as they schematized the mechanism, and showed that with biologically plausible, that is made up numbers that work, we could get a limit cycle. That is clearly a system that could, in principle, generate regular oscillations. And so the point here is, often when you're dealing with phenomena that are complex, especially in the way that oscillation is a complex phenomenon, one of the tools you need to understand how the mechanism is going to work is to represent it mathematically and show what you can get out of that mathematical representation. Now, the clock has more than one component, and people quickly started identifying, both in fruit flies and eventually in mice, a host of other components and began to give a story about where they might fit into the oscillatory system. And now you have the same old question. Will these extra components still leave a mechanism that could generate ongoing oscillations, or will this mechanism flatline? Well, Goldbeater said, I've got a lifetime occupation here. He has had he's done many other things as well. But he set out to say, okay, more components. This is now the sketch of the mammalian clock as of around 2000. Does the same thing, writes equations, shows that, in fact, under plausible parameters, they generate ongoing oscillations. He's able to start doing some more things. He's able to say, well, could we account for, in terms of this sketch of the mechanism, how the clock can be entrained to light-dark cycle in the environment, and so uses the model to show how entrainment works. Also began to think about pathologies of circadian pathologies. People like my spouse who finds bedtime and it can't be before about 5 a.m., and others who have to be in bed by 7 p.m. Those are circadian pathologies. Can you explain them in terms of how this mechanism might go wrong? And those are some of the things that Goldbeater continued to do. So what I'm drawing out of this first case is that we have a challenge of explaining. I want to move on first before I conclude that. Further problem. Everything I've told you about so far occurs within individual cells. But individual cells don't keep a very regular rhythm. They're variable anywhere between like 22 hours to 26 hours. But organisms keep regular rhythms. How does that happen? Well, people then turned their attention not to more details about the clock, but to the context in which these individual cells are working. That is, into a structure in the brain that had long been identified as likely to be important for circadian rhythms, the suprachiasmatic nucleus, and asked, well, how could a population of cells in the SCN generate regular rhythms? Regular, but it's not as if each one is pulsating together. This is an image from, is it not? It's not working up there, is it? Oh, it is? Okay, I'm not where I can see it. So you get a complex pattern of oscillation. This is a slice through the SCN using a fluorescent dye and showing that there's a pattern here. And the question is, well, now, how could these neurons that each of them have the capacity to generate rhythm work together to produce this kind of coordinated behavior? Well, there were some details about the components that were beginning to be helpful here. In the SCN, it's a bilateral structure, and each side has about 10,000 neurons. Some of them express the peptide vasoactive intestinal polypeptide, others of which express vasopressin, and some idea that we had two different networks that were somehow coupled. Question, how could you get a system like that to generate rhythms? Well, strategy came, let's do some modeling again. Can we propose ways this structure might be organized to see if we can see which ones would be ones that could actually generate this kind of rhythmic behavior? I'm not going through the details of any of this, but here's a highly simplified view by that time of what's going on within individual cells. All they've done is they've said, okay, it throws out a neurotransmitter, one of these peptides, and a receptor and a second messenger chain to connect up the different cells, and then see whether we can get the appropriate kind of behavior, and what we can then back infer about what might be the way the mechanism is organized to understand it. Notice I've been using words like might. This is how possibly, to use a phrase of Carl's. We might differ on how important how possibly is, Carl. To me, it's very important to see that these things are possibly able to do what we take them to be doing. So what I've been emphasizing then is computational modeling plays, I think, an important role in lots of mechanistic research, especially when it's not obvious from just looking at what the parts are thought to be doing, how this thing could produce the phenomenon in question. And so it allows us to then try to conceptualize the way the system works. So it's an indispensable tool, and it is continuing even at an accelerated pace because the number of problems in this domain has just continued to grow. First of all, we started with one. It was fun for me when we got to about 7 components. But now we've been able to show that about 1,000 proteins in mammals, modify them will either change the amplitude or change the periodicity of the rhythm. That is, all of these are affecting the rhythm. 1,000 proteins, folks, that's a good chunk of the whole population. And many of these have lots of other roles in the cell. We're not talking about a nicely segregated system. We're talking about a set of components that are highly coordinated and interactive with each other. That creates more challenges for modeling. When the research started on the suprachiasmatic nucleus, it was thought that's the locus where oscillations occur. Well, once you discovered the components of the oscillator, it was found that those are present in every cell of your body. They're expressed in every cell of your body. Every cell of your body is capable of generating circadian rhythms, and most do. But they don't do them all coordinated with each other. Rhythms or circadian oscillators in your liver are responding to conditions in the liver as well as maintaining a beat. And we thought at first, well, but they're all slaves. They just do what the SCN says. That's a bad way to think about the brain. We've discovered things go in both directions. And in fact, we're now realizing that things that perturb the circadian oscillators in liver cells or in kidney cells or in testes all feed back and have effects on what's going on in the SCN. I hope you appreciate it. We're now dealing with such a mammoth system. The only tools available to us are things like mathematical representation, certain visualization techniques, and so on, to try to understand this kind of a system. Okay, so that's case 1. Turn now to a very different kind of phenomenon, molecular motors, and a different lesson about energy. Too bad Larry Shapiro isn't here, for I have perhaps his favorite mechanism up there. But a point I just want to make.
Actually like bicycling.
No, I didn't think so. But you can do the other without bicycling. You need energy for any of these things to do anything. And so a question that should be asked of any biological mechanism is, where's the energy? And moreover, how do you go from free energy to the work that a mechanism is doing? Yes, how does free energy make things happen in mechanisms? So I've turned to a different example here. In the second half of the 20th century, a host of molecular motors have been discovered. This is a motor in cells, the myosin motor, the green component there, which goes through this dance of that Hugh Huxley told us about back in the 60s, of connecting up to the actin filament in the muscle, exerting a force on it. The biochemists came along, Lim and Taylor, and said, we got to connect this up to ATP somehow if you're going to get anything out of this swinging cross-bridge model, and proposed how the steps of ATP hydrolysis relate to the steps of myosin exerting force on actin. And yet that seems to leave a huge gulf. You've told us the energy enters here, but how is energy doing anything? And that led, at various points, that kind of thought led people to wonder, maybe free energy in ATP can't do this kind of work. There was an alternative model out there. Perhaps the movement comes from Brownian motion. And all free energy is doing is allowing for the binding of a ratchet in a Brownian ratchet kind of mechanism that's trapping Brownian motion. Andrew Huxley, famous to many people in this room for his work on good senior moment. Pardon? The action potential. Came over and was working on muscle and wondered whether there was any way to understand the movement of muscle other than perhaps drawing upon Brownian motion. 30 years later, Ron Vale similarly wondered, how in the world are you going to get ATP to generate motion? Now, shortly after Ron raised his worries in '91, people followed the same old strategy. Let's take myosin apart and see what its components are. Well, you can't literally take it apart, but you can develop tools like X-ray crystallography to find the structure of the molecule and then to ask, well, are there parts within it that might be doing different things? There's a site that was identified as the locus where ATP binds, another site where the myosin binds to the actin. But what people really caught on to was there was a structure over here that seemed to be a lever that was connecting up to the rest of the molecule. That might explain what you saw in that earlier image, the green object and the head swinging back and forth. So people were able to start to think, maybe what happens is this biochemical reaction of ATP hydrolyzing into ADP and PI is creating a little problem. You've got to have more space. It's pushing against the structure of the molecule. Descartes is really happy at this moment. It's push-pull mechanics. Pushing against the structure of the molecule, pushing then against the actin binding site, and pushing against the lever arm. And that idea bore fruition just a couple years later when people figured out how to. One problem with crystallization is you're taking something out in a very unnatural state to examine it. But people could try to find ways to have myosin bind to different mimetics and then crystallize it in different configurations. And by doing so, getting configurations that corresponded to being bound to ATP, then to ADP and PI, and to nothing, we're able to show that you had this obvious change of position in the as ATP was being hydrolyzed, and that distance, about 10 nanometers, corresponds to the distance that myosin was known to move along actin. So we start to have a story in terms of hydrolysis creating forces against other parts of the molecule that are intended to explain how you get motion out of hydrolyzing ATP. And notably, Ron Vale, just a couple of years later, says, oh yes, it's a power stroke. That's the language that's used here. We see how it works, except for a very crazy reference in a paper 3 years later. He never breathes the word Brownian ratchet, as far as I can tell. Now, Vale was actually perhaps better known for his work on another molecular motor, and this sort of is the parade case when you see people doing animations of molecular motors. He discovered that he was worried about the problem of transport along axons and what's called active transport, was looking at molecules that are associated with the microtubules that run along the axons, found one that hydrolyzed ATP, named it kinesin, and it's being illustrated there as lugging a big cargo, probably a mitochondrion, down the microtubule. And something notable that I'm not going to take time to develop today, but a lot of research went into now characterizing that phenomenon. That is a bad sketch of the phenomenon. But in 789, Vail did talk about kinesin walking hand over hand. But some mathematical work in the late 1990s said, you got a mathematical problem on your hands. The way you've described that mechanism, it's just going to wind up like a rubber band and stop when it's fully wound up. So that led people to say, okay, let's investigate this phenomenon further. Not the mechanism, just the phenomenon. How does it work? And came up with other ideas, like maybe it works like an inchworm, or maybe there's some other hand-over-hand motion. My graduate student, Andrew Bohagen, has done a very nice paper showing just what kind of research went into establishing that, yes, it does move hand over hand, but it's asymmetric, not symmetric hand-over-hand motion. Solved the problem, gave a model of the phenomenon. Other work that I'm not going into sketched where ATP is hydrolyzed in this mechanism and how it leads to the stepping motion of the molecule. I'm going to just note something very interesting about this work, and that is, as people identified kinesins, they discovered, oh, most of the time it's not doing this. Most of the time it's in this kind of configuration. And in that conformation, the two heads that normally would be binding the microtubule are off like this, and so you cannot walk along the microtubule. And it's only when something else happens that it puts it into the active form that you have kinesin able to walk. Then you get the kind of story that when there's cargo that activates the motor, the cargo attaches to the motor, gets a ride along the microtubule, leaves at its target location, and the kinesin either degrades or adopts this configuration again and hops on the back of another motor, the dynon, and takes a ride back to the center of the cell. Okay, so what's important here is often this mechanism is doing something very different. It's not moving cargo, but it's adopting a conformation where it can be floated through the cell and not consume ATP, because back of the envelope calculation says this guy would consume all the ATP of the cell easily. Okay, so what I'm pushed here is to understand how mechanisms perform work is important to understand that what they have to do is constrain free energy. And so we have to look for question to ask about any mechanism is, where is the free energy? How does it get there? But also, free energy flows. It needs to be constrained in order to do anything. What the components of the system are doing are constraining flows of free energy in particular ways. That's a retake on the idea that you have parts or entities and activities. You have entities constraining free energy to produce particular activities, and changing the organization can change the activity. So, sorry, Stuart, I used to believe in the Glennon law that there's one mechanism per phenomenon, but I think what the researchers are doing is showing us something different. Once they picked out a mechanism, they can take that as their reference point and start asking, does it do other things? And by the way, this was just one sketch of a case where the mechanism does more than one thing on different occasions. And now you've got a new question. Well, what determines what the mechanism is going to do? To turn to that, I'm going to turn to something I hadn't thought much about mechanisms before this, but you need to look outside the mechanism to ask what's going on around it, in particular, what's acting on it to make it do the things it's doing. And these are often going to be other mechanisms. So I find it useful to distinguish mechanisms that make products for the cell or the organism, things like the mechanisms of protein synthesis, and mechanisms that control those activities, what I'll call control mechanisms. And the basic idea comes from sadly neglected work of Howard Putty. To think about control, one needs to think about a mechanism that acts on another mechanism, so it's an effector, and does so in response to a measurement. Case of negative feedback at the top, measuring something that's produced by the system and using that to regulate the system. But also you can have forward control where you measure a condition in the organism or in the environment and use that to act on other mechanisms, changing what they're doing. So then the question is, what about this collection of control mechanisms? And I'm now turning to the nervous system. Well, actually, I will do a bridge before I get there. But the question I'm going to ask is how to think about this collection of control mechanisms. How are they organized? When we think about how to put production mechanisms together, or how to even put multiple control mechanisms together into larger mechanisms, this is what came up yesterday. We're talking about a part-whole relation, a compositional relation. That seems rather naturally to talk about it in terms of hierarchy. That was the terms that Simon and others brought to the floor. But when you turn to control, we typically do think, again, hierarchically. We think that there are the things that are being controlled, things above it that I'm using dotted lines to show getting information, making measurements, solid arrows to indicate producing effects, that we have controllers sitting on top of the controlled mechanisms, pulling the strings, if you will, to determine what they do. But biology doesn't like to follow our rules. Notice, this is how we've tried to organize our social institutions, often to failure, that putting somebody at the top isn't always the best way to get things done. But biology very much violates this, violates the hierarchy in all kinds of ways. First of all, sometimes you have controllers that are just off there on their own, exercising their control function, not listening to any of the others. You often have more controllers than things being controlled. And this is actually a gross underestimate. It's much, much more massive than this. Moreover, try to show it as levels, but very frequently you'll find things that you thought of as at one level controlling something at you thought a higher level. So the whole notion of levels breaks down, and there's nobody in charge, nobody at the top of the system. I dare say that despite the way we think about ourselves, that's a much better characterization of how our activities are being controlled. Most of us have come up to think about, well, if you think about brain, imagine most people's first association is the neocortex. Hey, wait a minute. Neocortex may be important, but there's a lot else to the brain. And a lot of people will say, well, you got the neocortex, and then there's the prefrontal areas of the neocortex, and they're in charge. And you got the central executive sitting up there in front. What I want to suggest is a brief look at subcortical structures gives you a very different understanding of the way in which control is operating in living organisms. Actually, you don't even have to go to the nervous system first. Just ask what goes on chemically to exercise control. The bloodstream is a potent way of distributing signals through the body. And so you have a really powerful set of control systems called endocrines. They were briefly mentioned yesterday, hormones. And here is just the case of insulin generated in particular cells, pancreatic beta cells, but acting on liver cells, friendly actin-myosin mechanism in muscle, in adipocytes, control of how glucose is used in these various components under chemical control. And that doesn't get supplanted when you add a nervous system on top. But what you get with the nervous system is lots of little controllers starting to play roles with this. But before I even jump to that, an important lesson from thinking about chemical control. Chemical control acts slower than the kind of electrical control that you commonly think of in the nervous system. Normally you think of transmitters released at synapses and picked up immediately at the synapse by inotropic receptors, creating a action potential in the recipient cell. No, slow it down. Chemical reactions occur over a slower period of time, and when you're packaged into neurons, the transmitters can be released at any point on the cell body, dendrite, or axon, typically not at the synapse, and they distribute by diffusion through the extracellular matrix to any other cell which happens to have a receptor for them. And often the receptors are G protein coupled receptors, which operate at least 2 orders of magnitude slower, that is 100 milliseconds or longer, rather than the second range for ionotropic receptors. So we've got a wholly different kind of slow information processing that can do things like set long-term agendas for the information processing by the faster systems. But the point here is it's organized in a really seemingly almost chaotic fashion. Signals coming up the vagus nerve, and I'm not going to be able to talk about it today, but one of my real favorites now is a part of the brain most of you have never heard of, the nucleus of the solitary tract, but into the signals also come up to the hypothalamus, to one structure in it, the arcuate nucleus, where you have a variety of neurons. I'm not going to try to give you the details here, but carrying out various assessments of how much energy do I have on board, how long has it been since I've had a meal, and should I be activating processes to get food, or should I stop eating? Or exogenic and exogenic neurons. The point here is you've got various local controllers carrying out specific tasks, but they often connect to each other. So let me look out a little bit broader at the hypothalamus. Well, it's the smallest part of your brain you can think of, but as was pointed out by a speaker not too many years ago at UCSD, you can live without your neocortex. You can't live without this guy. This guy's really in charge, but it's not a guy. It's a whole collection of guys and gals doing various different tasks. This is a schema of the hypothalamus. It's a bunch of nuclei, neurons in it, connect to each other, but have semi-specialized tasks. Arcuate nucleus, one of its tasks that I just discussed was measuring food content and whether you needed to eat. The suprachiasmatic nucleus right here, I mentioned earlier, center for circadian rhythms, and that's not the end of the story. I'm spending a lot of time now looking at the preoptic nucleus, which has major role in temperature regulation in the body, and this and point is I'm really trying to stress. These guys all connect to each other, coordinating activities, and with no one obviously in charge. And don't tell me that the cerebral cortex is actually in charge. No, it listens to these guys. And just to flesh that out, you find a set of neurons when these neurons in the lateral hypothalamus were discovered. They were named orexin neurons because they were thought to control feeding activity, but they also tend to be involved in maintaining arousal and a host of other activities. And they're pretty typical of neurons in various nuclei here. They get multiple inputs and they generate multiple outputs. So we've got really much more like a zoo or a network of interacting components. As Partridge in another ignored paper from 30, 40 years ago said, this is not the way an engineer would design it. But how about thinking of these guys as good enough calculi? They're good enough to get the job done, to do whatever job is needed to keep the organism alive. They've proven effective at maintaining the organism, even though doesn't make sense to us. That is, it doesn't give us a neat flow pattern of how information is being processed. Okay, so thinking about how organisms are, our mechanisms are controlled, what mechanisms do depend on, not just their parts and operations, but what's going on outside them, what's acting on them. I've emphasized this control doesn't seem to be hierarchical, creating this really very deep puzzle. How does hierarchical control work? How does it keep organisms operating and suffice to maintain organisms? So the perspective is there might be cobbling together over evolutionary time of good enough calculi that have enabled each organism in this lineage from wherever life began 3.8 billion years ago or so to now to stay alive long enough to reproduce. It's good enough to get the job done, and then we just keep adding kludges to that to keep getting the job done and not giving us a very intelligible idea of how the system is organized. Okay, so what's my conclusions here? There is a core idea about mechanism, and Stuart is, where's Stuart? Saw you earlier, there you are, has done a lot of work trying to articulate just what the common elements are, that we think that there are components that of a system that enable the system to do the phenomena that are associated with it. What I've tried to do here is say that we often need to do things like model the mechanism in order to understand how it might behave. We need to think about where free energy enters the mechanism and then think about the mechanism as constraining it. And we need to think about how these constraints within individual mechanisms are affected by other mechanisms so that at different times they do different things. And surely there's so much else. If I had another 30 years, I would hope to find, come back to you with another list of 20 new things about mechanisms that I do not know today. Because literally most of these things I hadn't thought of before I got into the various work I'm doing. So let me have a call to arms. Let's go see what's going on in biology. See what kinds of different things people are coming up with. And John did that with an example yesterday of different ways scientists are thinking about the explanations they're going to advance of given phenomena. Let's not go in with it has to fit a pattern, but let's find as much as we can about how it is that scientists are approaching these problems. And so a final note back to Bob's and my book, we were concerned at the time, we didn't want to just say mechanistic explanation is explanation. No, we wanted to figure out what would be an example of non-mechanistic explanations. And I think that's still an urgent calling. If Tony Shimero were here, I would apologize. Well, there you are, Tony. I don't know if you know that I have conceded on the question of whether the kinds of stories you tell are explanatory. Yes, they are. But you didn't convince me. I came along to see you were right because Art Winfrey convinced me. He gave me, you'll be happy to surrender that place to Art, who came up with a very nice argument. In a ocean, if there are tides, there has to be a place in the ocean where there's no tide. That's a mathematical point. He used it to say, if you've got a circadian clock that can show two different types of entrainment, there's got to be a light stimulus that will stop the clock. And he said, and I don't need to know the mechanism because we don't. This is 1986, and these guys at Brandeis hadn't come up with the TTFL at that point. So other kinds of explanations, and let's add to that. I mean, let's look at what people count as explanations. Okay, last note. I have lived my life benefiting from collaborators. Bob, you weren't my first co-author. Eric Stifler had that dubious distinction, but certainly a very major one. Many of you know Adele. Some of you know some of these other people up here. Point I want to make, I can say some of the things, I'm probably missing a lot of things, each of them has left on me. Ideas that I did not come up with, I got from them and have drawn upon them in elaborating my ideas. We've lived in a world in which we think philosophy is a solo practitioner's activity. I grew up in that world. My mentors at Chicago all published individually. No, it's a collaborative activity. And the more collaboration we do, the more we all gain from it. And so I have to thank Bob and all the other collaborators that came along to keep telling me, here's something new for you to think about. Thank you. Please.
All right.
I know there's a switch. Hello? No. Bottom? Bottom of my.
Did you figure out where the switch was on the mic?
There's a small red button on the bottom. Oh, on the bottom. All right, just a little. I like this though. But a little thing you emphasize, for example, systems in which there's no controller. And periodically you mention Simon. You really should mention the two together.
Mm-hmm.
And you even do other Simon-esque stuff sort of quietly on the side. I know Simon had lots of influence on you, and me. But one is that he emphasized that systems did not have hierarchical control. When it looked like there was a hierarchical control, usually the thing at the top that you thought controlled things did very little, and you could get rid of it, and the rest of the system would behave just fine.
Mm-hmm.
That was what he got the Nobel Prize for, theoretically. But it was just producing stuff that was good enough.
Yeah.
You kept putting it satisfying.
And of course, our whole notion of heuristic is a strategy that works well enough.
Yeah. I just wanted to tie Simon in more to your use.
Yeah. I can only acknowledge so many people in one talk.
Oh, Stuart. Yeah, thanks for that lovely talk. I wanted to ask you a question about control hierarchies and the relationship to compositional hierarchies,
Mm-hmm.
because it feels, you mentioned, when we were talking about the top-down causation thing, we were saying wholes can't cause parts, that whole thing. Whereas I think when you think about control hierarchies, they don't stand in those diachronic relations, or excuse me, they don't stand in those synchronic relations.
Yeah.
Right. And it is a little bit strange to think about them as part-whole relationships. And I guess one of the things that I'm maybe a thing I'm ultimately thinking in the back of my mind here is I think we don't have a clear enough sense of the relationship between part-whole relationships and these other kinds of dependency relationships in mechanisms. That's something we should talk more about.
I agree thoroughly. And here's, so some people have taken me to hold that an organism is a mechanism. Some of the people we visited in Exeter have held that view of us. I say, no, organisms are not mechanisms. They consist of a number of mechanisms often, but then we have to talk about the whole organization of the organism and how different kinds of relations between components in the organism enable each of its components to do the thing it needs to do, that the organism needs it to do. So something I'm really struggling with right now is to try and understand what's the relation between networks of controllers and organisms. How is it that, I'm working on trying to flesh out in an interesting way the notion of homeostasis that has come in for lots of criticisms over the years. But how is it that organisms maintain themselves through using mechanisms within them to do things and to control those doings of things? And I don't have good, conceptually, I haven't figured out how to hook those together. Likewise, how to think about relations other than compositional relations to think about putting things together. Clearly, your brain and your kidney are both parts of your body as an organism, but that doesn't seem to be very informative.
Right. And I think that part of the thing is that when we start thinking about things that we can think of structurally that are not localizable in obvious ways, and which also are the structures that are because of the dynamics. Then I think it, if you think about these particle relationships as blocks, then
You're dead.
you're dead, right? And yet that's, I think, I don't know what to say here. I just think it's something that really is something we should work more on.
So it's easy at times to put labels on things. I can put the word label hierarchy on this non-hierarchical notion, but I haven't I've told you what it's not, but I haven't told you a positive story of it. And I can, there's other terms I can throw out a term like homeostasis and start using it. But I think we really do, there's a lot of conceptual work to be done to understand what kinds of relations are important here toward understanding these systems.
Thank you. Yeah, thanks, Bill. That was super. Particularly, I really like, yes, the call to arms, the concession to Tony. I'm interested in a particular category of explanations and what you want to say about those. And I'm going to be talking a bit more this afternoon about your collaborations with Arne and Levy, talking about abstraction. So we've got mechanistic explanations of mechanisms, and then it looks like we've also got, let's say, non-mechanistic explanations, mathematical explanations, computational models, which do some explanatory work, but not by singling out mechanisms.
Well, some do and some don't.
Well, that's my question. So what do we want to say about computational models, which are actually intended to be taken literally as descriptions of physical computation? Do you think those are on the mechanistic side, the mathematical side, or a really interesting hybrid of the both?
Okay. First of all, I think we should not worry about, just as long as we're clear what we're meaning by the terms, there's not rigid rules. And so I'm not going to try to give you necessary and sufficient conditions for mechanisms. I think that's a fool's errand. But there is a point. Okay, so Adele and I, one of the last papers we did together, we're trying to understand what seems to be so different about computational modeling in the field of circadian biology and that which we had both grown up with and had both written about in cognitive science. We wrote a book together, two versions of a book together on connectionism. And it seems like there's something very different, that connectionist models were, in an extreme sense, a how possibly model. You could train a network and it will do things like humans do. And we'll call them neural, but those are not neurons. Those are not even Pitts-McCulloch neurons, let alone real neurons. So it seems like the modeling there is not grounded in trying to make sense of what we know about a mechanism and how it's doing things. It's rather trying to come up with some opening into a domain that can say, how is it that, I think one thing we learned from the earliest work on neural networks to today is we got really powerful pattern recognition systems. We can understand a lot about what it is to identify a pattern, even though now we've got ones that are so powerful we don't understand how they do it. But that's that's not, those models are not trying to describe the components of a system. Now, some people say, well, we are trying to do AI. We're trying to give artificial intelligence, and it might inform human intelligence. But we look at it and say, you're awfully far from describing any components of a mechanism. That's not a bad thing. I mean, it's a fine activity, but it's a different activity than when you already know some of the components and your problem is different. You don't understand how they could do the job. Now you're looking for job describers so that you can then look for who does that work. And an important point is the whole goal of putting out hypotheses in science is to learn how they're inadequate. If your hypotheses worked, you don't learn anything. It's when they rub up against nature in ways that say, you're failing to account for this. Now you've got some potential for insight.
Thank you.
Thanks, Bill.
Great talk. So I wonder if this notion of hierarchical, I want to hear more about it. I think it's a really neat idea. And I wonder if it connects up at all with, so Alcino Stillman and I last summer published a paper based on some new technology in molecular and cellular cognition, basically enable these mice to run around with 2 photon confocal microscopes on their head. And we suggest that instead of thinking upward and downward, think sort of inward to outward and back. So molecules, molecular pathways, inside cells, inside circuits, inside the behaving animal. And that seems to raise far fewer of these deep ontological questions about top-down causation and the like. And what is any kind of connection between what we were suggesting there and what you're suggesting with this hierarchical structure?
Ever since I gave a talk here, when you were still here, I've been rubbing up against, do I want to think in terms of levels? And Carl has been a very helpful agent in getting me away from most conceptions of levels and which ones work. And I get sucked into thinking in terms of levels when I think about control, because controllers act on something. It's natural to think of that as somehow a higher, a level relation. The problem is it breaks down almost at the next step. And then the question is how else to think about it. I mean, certainly I want to endorse in-out as a further thing, but also try to understand networks. And I spent enough time working with people in a network biology lab to realize there's a lot of resources there, but we really are scratching the surface of understanding how different organizations of networks can affect things. In individual cases, we can model it. I've come up empty-handed trying to say, what are the principles? Are there any design principles here? They don't have to be eternal. Just give me something. And this has been, for my whole life as a mechanist, I was worrying about organization. Can we, we can talk about discovering components. Can we say anything about organization? For years, I walked around saying, negative feedback, positive feedback. Can you give me anything more? And I actually sent then graduate student Marta Helena on a goose chase to try to find me some literature. I said, somebody has got to have thought about it. And actually, it was Arnon, when I was in Israel, told me about Yuri Alon's work. And I said, yes, you can think about these 2-3 component motifs and show what kinds of different properties they have. But what if you try to go to 4, 5? Oh no, this is all breaking down very quickly. So I don't get any more. I hope you see. I'm desperate for principles here. Not that they be eternal, but they are something we can use to go in and say, here's a starting point for trying to understand how this system does it. So we get a system that has lots of stages of going out to different ports. But can we might in an individual case be able to write a model that captures that system. But can we say, well, how could you modify that to do something else? And we're back at the drawing board.
Well, we don't purport to solve that problem, but we just sort of support to try to show a picture and how it's related to a development indirectly in science. That's all we're trying to do.
Yeah.
We don't have an answer to your question.
Yeah. Well, if you did, you would be somewhere else.
Sketching an image that might help.
Yeah. And I think, multiple images, none of which work perfectly, is perhaps the best we can do here.
So I really like this discussion about complicating the notion of mechanism. One of the things that's made me think that sometimes people get this really simplified version of mechanism as a single causal interaction between a certain set of components happening in sort of like almost a vacuum within a system, right? It seems like when we talk that way, it's almost as though the mechanism is supposed to carve up nature in some sort of really meaningful way.
Mm-hmm.
But when you start to say, look, we'll start with this idea of what the mechanism is, like in the circadian clock, and then you start to complicate it. It starts to become sensitive to background conditions or the initial conditions of the system. There are certain new kinds of inputs, outputs, and things like that. when mechanism plays that kind of role and it has more porous boundaries and things like that, it seems like it suddenly becomes a strategy for investigation or kind of idealization, but it seems to abandon that original way of talking where it seems to like cut up the world. Do you think that as we push mechanism to be sort of more open to these kinds of like larger system issues, that it changes the way we think about what role mechanism plays in the scientific process?
Okay. I think we have to walk back and forth along that path. Confession, I am not a realist. I'm much more concerned with the epistemology of inquiry than I am with any account of how the world is. Sorry, Carl, we part company on this issue. So for me, sometimes you want to come back to the narrow conception because you can do things with it. You can begin to work out things. And and what's an experimental technique but to cut things off from everything else and keep conditions constant? At least those conditions constant so you can observe these other changes. You're creating an artifact. You hope it's not totally misleading. You hope you can get enough mileage out of it before you concede that in fact you're falsifying things from the way they are in the living system. But I don't see any other way out of that, but we want to go back both directions. We want to recognize we have cut things off, and what we've cut off may just be what we need to know for the problem that we thought we could solve by cutting them off. Okay, so we went down that path, and now we have to back off and say, no, we learned a lot, but we didn't solve our problem. But you can't just open everything up. So at one point I wanted to say, echoing Bill Wimsatt, whose name hasn't come up yet today, but another powerful influence on my thinking, you must be a mechanist and a holist. And those are incompatible, but you need to be able to move back and forth, expand your conception of mechanism, but it's never going to become universal. But you might be able to pick up things that you need to account for, but then often to account for it, shrink back down and say, okay, now I'm going to take these components and treat them as if I can tell the story in isolation, only to see where I'm pushed back the other direction.
Well, yeah, Bill, that was a really beautiful talk, and thanks. It was great to listen to it. Since Stuart already asked this question and he didn't go to the point about Glenn's law, maybe I'll go there. I didn't see the conflict between you and Stuart, and so I want to let me articulate the way I see it. Then you're, I think, coming at it differently, and it might be an important way to get at the question of how we individuate mechanisms. So I take Stuart's point, which is related to one that Stuart Kaufman made as well. Be a point about how we individuate mechanisms. Mm-hmm. All mechanisms are mechanisms of something. And it's in reference to that we determine the relevance of the parts to the mechanism. And so calling it a law is kind of a joke, because it's not an empirical finding that for any mechanism there exists only one phenomenon for that.
It's definitional in a way.
It's rather something like, when we decide to take the mechanistic stance with respect to some system, we first specify the thing that we're trying to explain, and then what's in that mechanism and what's not, so it's relevant to that thing. And so in a way, we kind of stipulate from the beginning, this is the thing that we're concerned with. But then you said, aha, now I've been convinced that the same mechanism can do many things. And that suggests that you have a different way of individuating the mechanisms than by relevance to the phenomenon that they're getting at. And so I'm curious what your principle of individuation is, if not Glenn's law.
Okay. When Andrew and I were working on this, we started calling it Glenn's heuristic, and we figured Stuart would like to have it called a heuristic. That is, it's a fallible principle. It's a starting point. I don't know how else to start investigating a mechanism, but by figuring out what phenomenon it is that I'm trying to explain. But sometimes my collaborators have pushed things past me that they were right to do so, but I failed to recognize at the time. In the 2005 paper on mechanism, Adele said one or more phenomena. I said, no, it's got to be one phenomenon. But okay, marital harmony, we'll go with more. She's right. She's always right. But she was right here for sure, that in fact, after you've figured out the entities that you're going to think of as the mechanism, you can now, and I don't think it's us, it's the scientists, will sometimes latch onto them. The term mechanism goes with them as they start to probe what else this can do under different circumstances. So that you do get one mechanism, multiple phenomena. But usually, only after, you wouldn't typically set out saying, I've got 3 phenomena, let me find the mechanism for that. You start out by saying, I've got this phenomenon, this would explain it. Oh, but it's in this, these entities that I can now track are organized coherently to produce this phenomenon. But that very same coherent organization can be tweaked to enable the system to do other things. And I think we find this repeatedly in biology, that the biologists go the route of, okay, they don't care. But they'll say, here's the mechanism, and then they'll start talking about other things it does.
So is the principle something like same parts?
It is whatever you want to hold on to.
Same parts can, with a slight tweak.
Yeah.
Will do something that they didn't do before.
Yeah.
So what makes it the same mechanism is that it has the same components.
Same components. It's close enough. I think it's something. It's more like I can grab a hold of this somewhat. I can get enough of a grasp on it to start to do things with it. But I don't think there are good principles that absolutely individuate it. And I think that you make the decisions on the fly. What do I have to add this component in now that I didn't think was a component before? Not because it produced the first phenomenon, but because these other components work with it to produce this other thing. And throw the label. By the way, you don't usually see accounts of mechanisms in individual journal articles. Sometimes you do. Sometimes it's in the in the discussion section, and you'll get a figure, a cartoon of a mechanism. Mostly, it's in review articles and textbooks where people are trying to put the picture together. And there, I think they have moved away from the anchor point for the article, which was what was the phenomenon that I was getting data about, to what are these things that I think are real things in cells. I can be a realist that much. We think they're real things. So I think it's the practice goes in different directions depending on what works for the inquiry at hand.
Yeah, can I just follow up? So I guess one of the reasons why it's hard to decide whether or not it's sort of one mechanism, one phenomenon, we don't actually have principles for individuating phenomena. So is the phenomena A and B, or is there 2 phenomena A and B, right? And there's just an essentially stipulative notion there, and I don't think we should get too worked up.
We shouldn't get worked up. The scientists haven't got worked up.
No. But I saw 2 different things that you were doing. One thing you can say is that there are components that play some role in one mechanism that play roles in other mechanisms simultaneously, and that's
one thing.
What's going on in the cell all the time, right?
Yeah.
Another thing that you can say is that a certain kind of system which engages in behaviors have multiple kinds of effects that you could think of as, a car is driving along and it's generating heat, right? And we might have picked out for our pragmatic reasons the driving, but the heat's just as much a part of what the things do.
And if your muscles aren't generating heat, we got a dead mammal.
Yes, absolutely. I love the heat generating properties of my muscles.
Except under certain conditions.
Yeah. So it seems to me that we should realize that notion of phenomena itself is more labile than we sometimes give it credit for. that may account for some of this. Some of why maybe this is a distinction without a difference.
I think that, and there's a lot, when phenomena don't come prepackaged, so researchers investigate them, but they all have purposes and intents, and we got to look more broadly than the social is out there. I would put the social around everything, by the way, because I think it's also figuring out how we the physical doesn't come prepackaged. I mean, what we always were concerned about is there are things that, at least as far as we know, you can't change. And those are constraints on doing your mechanism. But the practice of science is a part of us, of a social enterprise, a political enterprise, and so on. And it's shaped by it. And we shouldn't get worked up about it, but we should try to figure out, how is it so damn successful? I mean, that's what I keep coming back to. This is a successful enterprise, and we want to understand the possibilities of success. If we have to violate Stewart's law to get something done, well, we'll do it. And he's not going to worry about it. If biological mechanisms were as fail-proof as
I wonder, as I listen to this exchange between you and Stuart and Carl in particular, where you emphasize a shift from one kind of mechanism, sort of one with hierarchical control, to one with hierarchical control, that if the real difference isn't just a matter of how much you emphasize organization. When you move to hierarchical control, you emphasize organization more than the things organized. And Stuart, rightly, in the cases he was looking at, emphasizes the parts.
Mm-hmm.
And the organization is there, but it's not crucial. I just wonder if this is, I like the idea of sort of these being continua on some kind of continuum, and they're really quite compatible.
Mm-hmm.
It's not likely to be
So I
trying to
can,
make peace.
yeah. I don't think, I mean, I think it's, so the anchoring points for thinking about mechanism, Stewart's minimalist notion, entities and parts, components, something, things, they're doing things, there's activities, work, whatever, operations, and they're organized. And for me, the weak point has always been organization, and that's where I've sort of been constantly, trying to make sense of what's the ways that you can take this stuff and organize it to do stuff. So yeah, that helps to explain. I would take the same components and watch the researchers show that different organization produces different things out of it. So for me, there's a lot hanging on the multiplicity of organization, whereas others are going to be more concerned with constitution.
Those are just matters of degree.
I think, oh yeah, and I think you can, and all of us will fall short and can look over to our friends and get some help.